Hello everyone, I'm Francesco and I'm a postdoc at the University of Sheffield in the group of Professor Heather Mortibois. And today I'm gonna talk about my poster entitled Testing iLCT Shortlisted Small Molecules in Parkinson's Patient-Derived Cells. So Parkinson's is a neurodegenerative disorder characterized by loss of dopaminergic neurons. And the main symptoms in Parkinson's are shaking, stiffness, difficulty with walking, balance, and talking. And the aim of this project is to test several drugs prioritized from the iLCT program in patient-derived cells. And we will test the effect of these small molecules on several parameters, such as mitophagy, which is the capacity of the cells to clear and temper mitochondria, autophagy, which is the capacity of the cells to clear and, in general, dysfunctional molecules, as well as mitochondrial growth and cellular morphology. The final aim is then to test these compounds and assess whether they can have a positive and beneficial effect on our patient-derived cell model. So for this presentation, I'm only going to show data on encyclinate, which is one of the several tested compounds selected to be tested in this project. So before showing results, I'm going to spend a few words about the methods. So our cell model consists in skin biopsies collected from both healthy control and patient with Parkinson's. From the skin cells, we have the so-called fibroblasts, which are cultured in flasks. Skin cells are then reprogrammed into stem cells. And in this specific case, we speak about iNPCs, that stand for neuronal progenitor cells. And the main advantage of this cell model is that they retain aging feature. Then we can differentiate these cells into neurons in a protocol that takes 27 days. And once we have the neurons, we can image them, we can stain these cells and then image using an high throughput microscope in order to obtain this picture that we can see here. Another few words on the staining. We can stain cells with different dyes, and these allow us to investigate different cells organelles, such as lysosome, as you can see here, stained in red, functional mitochondria stained here in orange, and total mitochondria stained in green. And then let's move now to the results. So as I said in the first slide, what I'm going to show today is some data about the encyclinate. So encyclinate is a drug that inhibits tumor growth and metastasis. And this is also an agent that causes mitochondrial depolarization in tumor cells. In Parkinson's, this drug has been shown to decrease the alpha-synuclein level, which is a protein that has a major cellular pathology, causes cellular pathology in Parkinson's. So as you can see in this slide, it's shown here, this drug ameliorates the defective mitophagy in the alpha-synuclein line in both the control cell line and in the patient cell line. Moreover, in sporadic Parkinson's, reduces the number of lysosome and also mitochondria, which is not shown here, but you can see in the poster. And so to conclude, we can say that in our lab, we develop a robust method to study neuronal disorders and neuronal diseases. These allow us to start from patient skin biopsies, reprogram skin cells into stem cells, and then differentiate them into neurons. And in the study, we aim to test several compounds from the iLCT program to assess whether these selected repurposed drugs can have a positive and beneficial effect on patient-derived cell model. And to date, some of the compounds tested are very promising. For example, encyclinate shows a restorative effect on some of the tested parameters, such as mitophagy index, mitochondria, and lysosomal number per cell. I would like to conclude to thank you all the people in Mortibois lab, to thank you Professor Heather Mortibois and Also, a good thanks to the founders of this research and especially the participants for donating their skin biopsies so this could happen. And thank you very much.